Scientists finally revealed the genetic origins of the Indo-Europeans. The results changed everything. For more than 2 centuries, one question has sat at the center of a debate that linguists, archaeologists, and now geneticists have never fully been able to settle. Where did the people who first spoke the ancestor of English, Hindi, Russian, Persian, and hundreds of other languages actually come from? Half the population of Earth today speaks a language descended from this single ancestral tongue, and yet nobody could point to the specific place, the specific population, or the specific moment when that language and the people who spoke it actually began. A massive new ancient DNA study, built from the genomes of over 400 ancient individuals and published by a team including Iosif Lazaridis, David Reich, David Anthony, and Nick Patterson, has finally located it, not somewhere in Europe. Not somewhere on the open steppe most people picture when they imagine the ancestral homeland. A narrow, specific corridor of land between the Caucasus Mountains and the lower Volga River, inhabited by a population nobody had properly identified as genetically distinct until this exact study pulled it out of the data. This debate is genuinely one of the oldest in modern historical linguistics, tracing back to the 18th century when scholars first noticed striking, systematic similarities between Sanskrit, Latin, Greek, and the Germanic languages, similarities too consistent and too deep to be coincidental, pointing toward a single common ancestral language that must have existed thousands of years before any of those languages were ever written down. Since then, candidates for the homeland of that ancestral language, usually called Proto-Indo-European, have included Northern Europe, the Balkans, Anatolia, and the Pontic-Caspian steppe, each with its own combination of linguistic, archaeological, and more recently, genetic evidence marshaled in its favor. The steppe hypothesis, associated for decades with the archaeologist Maria Gimbutas, and more recently refined by David Anthony himself, one of the co-authors of this exact study, had become the leading candidate among most specialists over the past 20 years, especially once early ancient DNA studies confirmed a major migration of steppe-descended ancestry into Europe around 5,000 years ago. What remained genuinely unresolved, even after that earlier wave of research, was where on the steppe, and among which specific population, this whole story actually started. Researchers call this newly identified population the Caucasus-Lower Volga cline, or CLV for short. And understanding why its discovery counts as such a significant breakthrough requires understanding what a genetic cline actually is, and why finding a new one, sitting in exactly the right place at exactly the right time, is such a big deal. A cline, in population genetics, is a gradient, a spread of individuals whose ancestry shifts gradually and measurably as you move across it, rather than a single uniform population all carrying identical DNA. Researchers building this study assembled whole genome data from 372 newly sequenced ancient individuals, dating across a span from roughly 6,200 to 2000 BCE, combined with almost 1,000 previously published genomes, quadrupling the amount of Yamnaya period data available for analysis, and roughly doubling the sample size from the broader Neolithic period immediately before it. That scale mattered enormously, because it let researchers see something that smaller, earlier datasets simply hadn't had the resolution to detect. Not one, but 3 separate, previously unrecognized genetic gradients sitting in the region just north of the Caucasus and along the Volga and Dnipro rivers in the centuries right before the Yamnaya culture exploded outward across Eurasia. The first of these 3 gradients researchers identified runs along the Volga River itself, oriented roughly north to south, with individuals arranged almost perfectly according to where along the river they actually lived. At one end sit Eastern hunter-gatherers, a population spread across a broad stretch of territory well before farming or herding reshaped the region. Moving down the gradient, you find people connected to the Khvalynsk archaeological culture, and further along still, at the southern end, individuals from a site called Berezhnovka in the lower Volga, whose genetic profile closely resembles material from other sites called Progress 2 and Vonyuchka, found further south in the North Caucasus. Researchers call this the Volga cline, and it turns out, despite its name suggesting it should be central to the Yamnaya story, to be essentially irrelevant to how the Yamnaya culture actually formed. The second gradient runs along the Dnipro River further west and represents a different mixture entirely. Local Ukrainian hunter-gatherers from the Dnipro basin, mixing to some degree with hunter-gatherer populations from further east along the Don, and eventually connecting to individuals from the Sredny Stog archaeological culture. Researchers call this the Dnipro cline, and while it also turns out not to be the direct source of Yamnaya ancestry, it plays an important supporting role in the larger story because the Yamnaya themselves turn out to sit as a kind of extension of this exact gradient, a population that formed as what researchers describe as the late Sredny Stog population, directly descended without further mixture from an earlier point along the same Dnipro line. The third gradient is the one that changes everything, and researchers named it the Caucasus-Lower Volga cline, specifically because it runs between those 2 points. Berezhnovka at one end, in the Lower Volga region, and at the far southern end, Neolithic populations from the South Caucasus, at a site called Akhnishen in what is now Armenia. In between sit intermediate populations, including one from a site called Progress II in Vonyuchka in the North Caucasus foothills, another called Remontnoy that sits genetically between the northern and southern extremes, a material connected to the Maikop archaeological culture, a Bronze Age culture long recognized by archaeologists as culturally significant but never fully understood in terms of exactly how it connected genetically to the populations around it. This CLV cline, researchers found, persisted for roughly 1,000 years, from about 4,500 to 3,500 BCE, and it turns out to be the population that everything else in the story actually grew out of. Here's where the study's central discovery comes into sharp focus. The Yamnaya culture, the archaeological horizon associated with massive Kurgan burial mounds and wagon-based pastoralism that appeared around 3,300 BCE and, within a few centuries, spread from Hungary in the west all the way to Kazakhstan in the east, has long been recognized by geneticists as the population responsible for spreading steppe ancestry, and quite possibly the earliest widely spoken Indo-European languages across an enormous stretch of Europe and Asia. What hadn't been clear before this study was exactly where the Yamnaya themselves came from genetically before they existed as a distinct, identifiable population. This research answers that question with real precision. The Yamnaya can be modeled as descending from the CLV cline with essentially no additional outside mixture, drawing roughly 80% of their ancestry, 4/5 of the total, from the Caucasus-Lower Volga population specifically, with the remaining 20% coming from Ukrainian Neolithic hunter-gatherer ancestry connected to that separate Dnipro-cline discussed earlier. That finding alone would already represent a major clarification of Yamnaya origins, but researchers went further, using a technique that lets population geneticists estimate not just what a population is made of, but roughly when that specific mixture first formed. When two previously separate ancestral populations begin having children together, the DNA each child inherits comes in long, continuous chunks from each parent population, chunks that get progressively shorter and more fragmented with each subsequent generation, as ordinary genetic recombination shuffles chromosomes a little further apart with every new pairing. By measuring the average length of these ancestry blocks in the Yamnaya genomes available, researchers could date the specific mixing event that created the Yamnaya population with real statistical precision, placing it at approximately 4,100 to 3,900 BCE, directly in the same window when the broader CLV cline was already active and interacting with neighboring populations. The genetic data also revealed something about the scale of the specific group responsible for this formation, and it's a detail worth sitting with because it reframes how you should picture the whole event. Rather than a large, established population simply drifting into a new cultural identity, the data shows what researchers describe as an intense founding event, a relatively small number of individuals, likely just a few thousand people at most, who then expanded with extraordinary speed into a population of tens, and eventually hundreds, of thousands, within a remarkably short span of archaeological time. That's the genetic signature of a small, tightly connected founding community, quite possibly built around a specific innovation in culture or economy, whose descendants then multiplied and spread outward fast enough to reshape the genetic landscape of an enormous stretch of the ancient world. The earliest population researchers could identify as fully genetically Yamnaya, in this bottlenecked sense, comes from a site called Mikhailivka in the lower Dnipro region, dated to roughly 3,600 to 3,300 BCE through radiocarbon analysis, sitting in one of the rare parts of this broader region that shows continuous settlement spanning the transition from the Neolithic into the early Bronze Age, making it a genuine candidate, though researchers are careful to describe this specific point as speculative rather than confirmed, for the actual place where the Yamnaya identity as a distinct population first took shape. Once formed, this small founding population expanded so explosively and so fast that it left behind a genuinely unusual genetic signature, one that surprised the research team even though they were the ones who found it. Normally, when a human population spreads gradually across a large territory over a long period, you'd expect a clear, measurable relationship between physical distance and genetic relatedness. People buried closer together tend to be more closely related than people buried farther apart, because the population had time to build up local genetic structure as it spread. The Yamnaya genomes examined in this study barely show that pattern at all. People buried in cemeteries a few hundred kilometers apart turn out to be only marginally more closely related, genetically, than people buried thousands of kilometers apart across the full breadth of the Yamnaya cultural horizon. That's the genetic fingerprint of an expansion that happened too fast for normal geographic genetic structure to build up behind it, a population moving and mixing across enormous distances within a handful of generations, effectively outrunning the usual pace at which genetic relatedness would normally track physical geography. The CLV Klein's influence didn't stop at producing the Yamnaya, and this is where the study's findings become genuinely global in scope, rather than a story confined to one region north of the Black Sea. Migrants connected to this same CLV population moved outward in multiple directions simultaneously, leaving detectable genetic traces in several different, previously puzzling populations across a wide stretch of the ancient world. Some CLV migrants moved a very long distance westward, bypassing established intermediate farming populations like the Cucuteni Tripilia culture entirely, and turning up essentially unmixed in the genomes of individuals recovered from sites in what is now Croatia and Hungary, evidence of direct, long-range westward movement rather than the more gradual, step-by-step diffusion researchers might otherwise have assumed. Other CLV migrants moving in the same general westward direction did pick up some admixture along the way, mixing modestly with Tripilia populations to help form the later Usatov culture, and contributing in smaller measure to still other regional populations further west again. The most genuinely unexpected finding in this entire body of research, though, involves a population much further south, in central Anatolia, in what is now Turkey. Researchers examining Bronze Age genomes from sites called Alaka Hoyuk and Arslan Tepe, spanning roughly 2800 to 1500 BCE, and associated archaeologically with populations likely to have spoken Anatolian branch languages, including Hittite, one of the oldest attested Indo-European languages in the historical record, found that these individuals carry a substantial share of CLV-related ancestry, mixed specifically with populations connected to ancient Mesopotamia. That mixture wasn't a minor genetic footnote either. Depending on the specific modeling approach used, CLV ancestry accounts for somewhere between 10 and 30% of these individuals' genomes, and the remaining majority, in some individuals as much as 70 to 90%, traces not to the local farming population that had lived in central Anatolia for millennia beforehand, but to incoming Mesopotamian-related ancestry instead. That combination represents what researchers describe as a hard population replacement, a population that looks, on the surface, similar to its geographic neighbors, but that turns out, once examined in genetic detail, to be almost entirely discontinuous with the population that had lived in that same region immediately before it. Earlier research on these same specific sites, including the original study that first published this material in 2018 and a follow-up reanalysis in 2022, hadn't detected this pattern at all, underscoring just how much additional resolution this new, much larger dataset was able to provide. That discovery matters enormously for one of the oldest disputes in historical linguistics. Whether the language family researchers call Indo-Anatolian, the common ancestor of both the separate Anatolian branch, including Hittite, and the much larger Indo-European branch that eventually produced everything from Sanskrit to English, actually originated somewhere in the west, spreading into Anatolia from the Balkans, or somewhere in the east, spreading in from the Caucasus region instead. For a long time, the dominant argument favored a western route, based partly on the observation that Anatolian branch languages show an entirely western geographic distribution in the historical record, and partly on the argument that a route arriving from the west offered a simpler, more direct explanation for that distribution than a more convoluted eastern path would, since a straightforward westward to eastward line of settlement naturally produces a cluster of related languages sitting toward one side of the map, rather than scattered unpredictably across it. This new genetic evidence pushes hard in the opposite direction. The population responsible for likely bringing Anatolian languages into central Anatolia doesn't show any of the specific Anatolian farmer or Balkan Copper Age ancestry a western route would have been expected to leave behind along the way. Instead, it shows a genetic signature connecting directly back to the CLV Klein and to Mesopotamia, evidence pointing toward an eastern route into Anatolia, rather than a western one, a genuinely significant reversal of what had been the more widely favored explanation among linguists working on the specific question. Researchers studying this puzzle proposed a specific explanation for why Anatolian languages ended up with an exclusively Western historical distribution, even if their deeper roots trace to an Eastern route, a separate, well-documented population movement called the Caucasus expansion, occurring around 3000 BCE, which the same research team had previously shown in a 2022 study involved a complete, hard replacement of the earlier Caucasus-associated population in that specific region. That later expansion may have driven a wedge of speakers of an entirely different language family, possibly ancestral to Hattic or Hurrian languages known from the historical record, directly through the middle of the territory where Anatolian Indo-European related languages had previously been spoken, on both the northern and southern sides of the Caucasus, effectively erasing any trace of earlier Anatolian related language use in the eastern part of the region, and leaving behind only the western distribution later historical sources actually recorded. It's a proposed explanation rather than a fully settled one, and the researchers themselves describe the case for a western route as still plausible, just now considerably less favored than it was before this specific genetic evidence became available, something they estimate shifted the balance of probability from roughly 5 to 1 in favor of a Western route, based on the linguistic arguments alone, to roughly 5 to 1 in favor of an Eastern route, once the genetic evidence is factored in. If the CLV cline really does represent the population responsible for the common ancestral language behind both the Anatolian branch and everything else in the Indo-European family, that raises an obvious follow-up question. Where, within that population, did later, more specifically, Indo-European branches, the ones that eventually produced Indo-Iranian languages, Sanskrit, and dozens of other major language families, actually originate? The genetic modeling offers a specific, testable answer here as well. Since the Yamnaya themselves derive roughly 80% of their ancestry from the lower Volga end of the CLV cline specifically, and since the Yamnaya are widely understood by researchers as the population responsible for spreading what's sometimes called core Proto-Indo-European, the branch of the language family that excludes only the earlier diverging Anatolian languages, it follows fairly directly that if the lower Volga population spoke an early Indo-Iranian related language, the Yamnaya would simply have inherited that same language along with the bulk of their genetic ancestry. Genetics on its own can't fully settle whether the deepest linguistic roots trace specifically to the southern, more Caucasus Neolithic end of the CLV cline or the northern, lower Volga end, since both ends belong to the same broader ancestral gradient, rather than being cleanly separable populations. Resolving that particular detail, researchers note, will require synthesizing the genetic findings with archaeological and linguistic evidence together, rather than relying on ancient DNA alone, including specific clues like shared cereal farming vocabulary across different branches of the Indo-Anatolian family, evidence some linguists have pointed to as support for a homeland where agriculture, rather than pure pastoralism, played a meaningful role in daily subsistence, a detail that would fit more naturally with the southern, more settled end of the CLV gradient than with the more mobile, herding-focused populations further north along the Volga. The final major thread of this research moves the story forward in time and geography, from the Yamnaya heartland into Central Europe, addressing a separate but closely related puzzle: exactly how steppe ancestry, and by extension Indo-European language, spread into the population responsible for the corded ware culture, the broad archaeological horizon that swept across large parts of northern and central Europe, beginning around 2900 BCE. Researchers found that the specific steppe ancestry present in corded ware genomes traces overwhelmingly back to the Yamnaya specifically, sharing the same distinctive genetic founder event dated to roughly 3600 to 3400 BCE that defines the Yamnaya population itself, rather than to some separate, independent, steppe-related source population that some earlier research had proposed as a possibility. The way that ancestry actually moved into corded ware communities turns out to be genuinely more complicated and more interesting than a simple story of steppe migrants displacing and replacing the farming population they encountered. Examining a specific early corded ware site, researchers found individuals ranging across the full spectrum between pure Yamnaya-like ancestry at one extreme and pure globular amphora farmer-like ancestry at the other, with most corded ware individuals settling somewhere in the middle within just a handful of generations. Crucially, when researchers looked specifically at which individuals sat at each extreme of that range, a clear pattern emerged. Every individual at the fully Yamnaya-like extreme and every individual at the fully globular amphora-like extreme was female. That pattern points toward a genuinely different mechanism than the traditional, more simplistic assumption that steppe expansion into Europe was primarily driven by migrating men, an assumption that had shaped a lot of earlier popular thinking about how steppe ancestry spread west in the first place. Instead, the evidence suggests women connected to both the Yamnaya and globular amphora populations were being incorporated into corded ware communities in successive waves, gradually reshaping the broader corded ware gene pool while, on the paternal side specifically, the population continued to carry Y chromosome lineages connected to the original, earlier corded ware paternal line, a lineage genuinely distinct from the specific paternal lineage most strongly associated with Yamnaya men. Researchers involved in this analysis were careful to note that this doesn't necessarily rule out male-driven dynamics playing some role earlier in the broader steppe expansion story either, since it remains entirely possible that Yamnaya or globular amphora men were, in some contexts, taking multiple wives from these other populations, producing exactly this kind of asymmetric pattern without requiring a single, simple story about which sex drove which specific stage of the overall migration. That combination, a female-mediated genetic shift running alongside a comparatively stable paternal lineage, offers a real, evidence-based resolution to a puzzle that had genuinely confused researchers working on this specific question for years. How could corded ware populations carry so much Yamnaya-derived ancestry overall, while so rarely carrying the specific Y chromosome lineage most closely associated with Yamnaya men themselves. It's also a good example of how ancient DNA can correct assumptions that earlier, less detailed research had simply taken for granted, since a full genome carries a mix of both maternal and paternal ancestry, while Y chromosome data alone only tells half of that story, and relying on paternal lineage data by itself, as a lot of earlier steppe migration research had to, risked producing a misleadingly simple picture of how these populations actually formed. Taken together, this body of research answers several genuinely distinct questions that had each, on their own, remained unresolved for a very long time. And it does so by finding new structure in exactly the window of time and geography where the answer had always logically needed to be sitting, just beyond the resolution of any previous, smaller dataset. It identifies, for the first time with this level of confidence, 3 separate genetic gradients active in the region north of the Caucasus and along the Volga and Dnipro rivers during the centuries before the Yamnaya culture existed as a distinct entity. It shows that one of those 3 gradients, the Caucasus-Lower Volga cline specifically, functioned as the genetic wellspring for the Yamnaya themselves, contributing roughly 4/5 of their total ancestry in a mixing event precisely dated to around 4100 to 3900 BCE. It traces migrants from that same CLV population outward in multiple directions, including a long-distance eastern route into central Anatolia that offers new, genuinely significant evidence for where the shared ancestral language behind both the Anatolian and broader Indo-European branches most likely originated. And it resolves a long-standing puzzle about how Steppe ancestry actually flowed into the corded ware culture of Central and Northern Europe, revealing a genuinely unexpected female-driven pattern of genetic transmission running alongside the population's more visibly stable paternal lineage. None of this fully closes the book on the Indo-European homeland question, and the researchers behind the study are careful to say so themselves. Genetics alone cannot specify which precise dialect or language variety was spoken by the CLV population, or resolve every remaining disagreement between the linguistic and archaeological communities about exactly how and when specific branches of the family split apart from one another. What it does provide for the first time is a genuinely direct, physically grounded anchor point for a debate that had spent more than 2 centuries relying almost entirely on comparative linguistics and archaeological inference alone, without any way to test those inferences against the literal genetic record of the people who actually lived through the events in question. It's worth appreciating just how much this specific study had to get right simultaneously for its conclusions to hold together as well as they do. It needed a large enough sample size to detect genuinely subtle population structure that smaller studies had missed entirely. It needed precise enough radiocarbon dating to place that structure in the correct chronological sequence relative to the Yamnaya expansion. It needed sophisticated enough statistical modeling to distinguish real ancestral mixing events from the kind of superficial genetic similarity that can mislead researchers into drawing the wrong conclusion. Exactly the trap that caught the original 2018 and 2022 studies on the central Anatolian material, before this larger dataset revealed what they had missed. And it needed researchers willing to synthesize genetic findings with linguistic and archaeological evidence honestly, acknowledging where the DNA alone can settle a question and where it genuinely can't, rather than overstating what population genetics by itself is able to prove. That combination, methodological rigor paired with real intellectual honesty about the limits of the evidence, is part of why this specific study has been received as such a significant, credible advance rather than just another entry in a long list of competing homeland theories. A narrow strip of land between the Caucasus Mountains and the lower Volga River, inhabited by a population that stayed distinct and interconnected for roughly 1,000 years before exploding outward in every direction at once, now stands as the best supported physical starting point science has ever been able to offer for one of the largest, most consequential language families in the history of the human species, a family whose descendants, more than 6,000 years later, are spoken today by close to half the people alive on Earth. What started as a small, tightly bottlenecked community, quite possibly numbering only a few thousand people at its founding, expanding fast enough across the ancient world to outrun its own genetic geography, ended up shaping the words spoken in homes, marketplaces, and governments from Iceland to India, and it took over 400 ancient genomes and more than 2 centuries of accumulated linguistic and archaeological groundwork before anyone could finally trace that story back to the specific stretch of ground where it actually began. And there is one final reason this discovery changes the way we should think about the Indo-European question. The story is no longer simply about choosing between competing dots on an archaeological map. It is about reconstructing a sequence of relationships that unfolded through time. The CLV cline gives researchers a population framework, the Yamnaya gives them a later expansion, and the genomes from Europe and Anatolia provide evidence of how different branches of that expansion may have interacted with populations already living there. Each piece becomes more meaningful because it can be compared with the others instead of being interpreted in isolation. The next stage of research will likely depend on finding more ancient individuals from the regions between the known sites. Every new genome can potentially narrow the gaps between populations that currently appear separated in the record. More precise dates could also clarify whether movements happened as a single episode or through several overlapping waves. Archaeological discoveries may reveal whether the genetic transitions corresponded to changes in settlement patterns, burial customs, mobility, farming, or herding. Linguists can then test whether the reconstructed vocabulary fits the environments and economic practices suggested by the archaeology. None of these disciplines has to provide the entire answer by itself. The strength comes from seeing whether independent evidence points toward the same sequence. A population that was difficult to recognize only a few years ago can become visible when researchers add enough genomes and apply better models. A migration that once seemed hypothetical can become genetically measurable, and a debate that lasted for centuries can suddenly move from broad speculation toward a much more specific geographical and chronological framework. The mystery is therefore not completely finished, but it is no longer as mysterious as it once was. The evidence now points toward a real population landscape, a real period of interaction and a chain of migrations whose consequences reached far beyond the communities that first experienced them. Somewhere in that ancient landscape, people lived ordinary lives without knowing that their descendants would become part of a story stretching across continents. Their bones preserved the evidence. Thousands of years later, science finally began reading it,